You know, I have to adjust that. That's the usual exercise. Sorry for that. Again, good morning to everybody, and thanks to the organizer to invite me to this outstanding conference and to this outstanding place. It's a great pleasure to be here at ICRISAT and to celebrate together with you the 10th anniversary of the Genomics and in Future Systems Biology Center, which really has set a landmark regarding the genomics and the exploitation of genomics of tropical legumes. And I think this place here is unique in a sense that research at ICRISAT covers the entire value chain of plant genetic resources, from their collection, their storage and multiplication in gene banks, research up to downstream plant breeding and securing of food security. And there are few places in the world which are so excellent in that regard, like this place here. And this, of course, is also something, this topic, the value chain of plant genetic resources, is also a topic that we at IPK-Gatersleben are deeply pondering about. And this is also the context, of course, of but this will be the context of my presentation today, which lists the co-authors here who contributed to the results and the ideas I'm going to present in the minutes. Well, if we think about the value of a plant genetic resource, we could describe it in a simple equation or in simple terms, that the value is a function of the quality of a genetic resource, the quality of a seed sample or of a plant or tuber sample, their availability within an ex situ gene bank or from ex situ gene bank, and of course the information that we can link to the biological or physical genetic resource. And in a snapshot, I think gene banks have done a very good job in terms of the representation of and representativeness and completeness of their collections, the seed quality, which is secured by regular multiplications of the material, and of course the availability of ex situ material, which with many gene banks, unfortunately not all, but with many gene banks delivering their material and filling in the requests basically 7 days a week, 24 hours a day, and 365 days a year. However, the black box that concerns and that attracts our interest is the information on the material that we store in our gene banks. And there are two types of information, as you know, of course. That's phenotypic information and genotypic information. And starting with phenotypic information, most gene banks have what you could call a hidden treasure. They have a large body of characterization data. And what are characterization data? These are data that are regularly collected by the curators of the collections during the multiplication process, and they are being recorded in order to make sure that the authenticity of the samples is correct. And most of these characterization data are morphological descriptors that have been defined by international panels for each individual crops. Many of those descriptors are, let's say, of academic value and not really of any impact for the genetic improvement of a crop for plant breeding, in other words. But some of those characterization data also are fairly useful for plant breeding. And in case of the barley and the wheat collection in our gene bank at IPK, it's characterization data that are regularly captured with each multiplication step or cycle on 1,000 grain weight, on flowering time, and on plant height. Now, that at first approximation sounds good, but there is, of course, a caveat to those data. As you know, gene banks no longer multiply their material each season or each year, since we store it at minus 10 or minus 20 degrees centigrade. We multiply the material in case of Gatersleben, for instance, only about every 40 years regarding the wheat collection, for instance. That means the data points we have are for each accessions are limited, and the experiments are not randomized experiments that can be easily analyzed and evaluated. Nevertheless, there are approaches, and one such approach has been published a few years ago, which analyzed the available characterization data of about 10,000 wheat accessions that were multiplied in the field at these intervals of about 20 to 40 years between the years 1946 and 2009. And what you can see here is that using this non-redundant product method is non-redundant ranking procedure, that this procedure helps to order the accessions according to their trait performance on a normalized scale between 0 and 1. And fairly high heritabilities can be achieved for their trait. So these legacy data, they represent a fairly good predictor of the actual performance in the field regarding those traits of the material that we keep in our gene bank shelves. And just remember that, for instance, climate change and agricultural practices have changed a lot during these more than 50 years. And therefore, it's a true challenge to use those legacy data to make predictions, let's say, into the future. And Jochen Reif and his research group, they are presently working on further improving the analysis of these legacy data to achieve even a higher accuracy regarding the comparison between legacy data and the actual performance of these genetic resources in the field. So this is a true treasure grove which has which can potentially have a high impact for plant breeders. Nevertheless, as a research institute, we are of course also very interested to de novo capture phenotypic data and to further resolve trait-related data, agronomic trait-related data into their components. And for that reason, we have been establishing, similar to other research institutions, few other research institutions worldwide, a technical infrastructure that will allow us for phenotyping and for using various technologies to phenotype plants from the cell level, from the analysis of individual organs, individual plants, mainly in greenhouses and growth chambers, up to monitoring plant growth in the field. And a recent addition to this technological infrastructure was the construction of what we call a plant cultivation hall. That's a huge phytotron with more than or with about 1,000 square meters area that can use to grow plants under highly defined environmental conditions that even may allow projections regarding future climate and weather conditions. And this infrastructure is part of a larger European research infrastructure that is indicated here. Right, so one simple example of using one of those infrastructures is imaging-based phenotyping in the context of genome-wide association analysis, which we performed in this specific experiment for barley. We were interested to analyze digital or to analyze biomass development on a daily basis and under control and under drought conditions, and basically to run a genome-wide association analysis for every day of the experiment. And what you can see here on this slide is that when we subjected this mapping panel or this GWAS panel to a drought phase during the experimental day 27 and 46, we see two types of SNP associations coming up. There are markers or there are marker trait associations that become significant during the late drought phase mainly. And then there is an additional set of markers that become significant and show significant effects during the recovery phase. So this gives us a first a possibility to put our fingers or to have, to start to peel the onion of this complex trait, drought tolerance, and this imaging-based technologies which allow us to have a temporal resolution of GWAS and the effects of individual SNP markers allows us to uncover and unveil the choreography of QTLs during plant development in a non-destructive manner. Right, just another sample. Well, I should say this kind of experiment and analysis using this imaging-based phenotyping in conveyor belt greenhouses is limited due to the limited throughput presently on the analysis of core collections that may comprise, for instance, 200 to 300 individuals or genotypes. So this is still way off an entire gene bank collection. Nevertheless, there are other technologies around that would allow for and that do allow for the systematic screening of large gene bank collections. And one such example is given here. This is a high throughput NMR-based screen to analyze the lipid content and lipid distribution in oat grains. That was performed in this study, which has been published back in 2014 already. And in this study, within a fairly short or brief period of time, the entire oat or Avena collection of our gene bank, and that comprises more than 3,000 accessions, has been phenotyped using this technology, nuclear magnetic resonance. Well, so that just should have given you, or that these two examples just should give you a glimpse or an idea of where we move to regarding phenotypic and trait analysis of gene bank material. And now we will shift gears and move on to genotypic information. Well, genotypic information is at the heart of that meeting, and it has been mentioned already before. Many, if not most, crop plant genomes have been sequenced in the past 20 years or so. Some of the crop plant genomes, or most of the crop plant genomes, are fairly complex. We do not have finished templates or finished reference sequences of those genomes, but at least we have draft genomes available. In terms of barley, wheat, rapeseed, and rye, our institute has been fairly active over the recent years, and one of the motivations to work on resequencing or on sequencing crop plant genomes is that we expect benefits both for gene bank management as well as for the utilization of plant genetic resources in terms of research, and finally, of course, in terms of breeding new cultivars, which will be done by plant breeders at the end of the day and not by our institute. Well, major emphasis and most dearly to our heart has been the barley genome, which has been the sequence of which has been initiated by the International Barley Sequencing Consortium back in 2007, a time when next generation sequences have proven applicable also to look into complex serial genomes. And well, the most recent outcome of this research is now the high quality reference of the barley genome. I think that's one of the best reference sequences of a crop plant genome, which is available these days. And this reference sequence allowed us to gain a lot of insights, of new insights into the evolution of the barley genome and into the impact of barley breeding and, of course, also barley domestication on genome diversity. But to get real deep insight into what's happening and happened in terms of the evolution of genome diversity, we need to start to sequence and resequence systematically the entire barley collection of the Institute, and this is one of the largest gene bank ex situ collections worldwide, amounting to almost 24,000 accessions. And those 24,000 accessions have been resequenced using genotyping by sequencing in a project that has been initiated about 2.5 years ago. Well, and one of the most immediate spin-offs and outcomes of such a resequencing exercise in terms of gene bank management is the issue of the identification of duplicates in our gene bank collections. And what you see here is that, or what you probably know is that there are about 7 million ex situ accessions worldwide in our gene banks, and according to FAO estimates, this explains or contains an inherent redundancy between collections of about 3.5-fold. That means the effective population size is approximately 2 million ex situ accessions worldwide. Nevertheless, there would be good reason to have a redundancy within the system because this provides additional security. This could provide improved access if an accession is available from several gene banks, and since all gene banks or several gene banks would collect data on a specific accession, this would also increase the level of phenotypic information. So by and large, this is a good thing to have a redundancy between gene banks. However, it increasingly turns out that there is also redundancy within collections. And the redundancy within the collections is of unknown extent. And of course, this is a nuisance because it would be a depletion of resources if we preserve and multiply material of identical accessions. Well, and in a first approach at this duplication issue, we were looking at passport data from our gene bank documentation system at IPK. And well, to sift these list of passport data, and this is just a random screenshot of the passport data of barley, we see that there is up to 28% here on this screenshot of potential duplicates just based on the passport data. Now, if we now complement this with the GWAS resequencing data of the first approximately 15,000 samples that have been resequenced to the date of this analysis, we learned that this sample of 14,400 accessions had some 4,400 potential duplicates based on identical GWAS fingerprints. So the effective population size would be close to 10,000 samples only, and not 14,400. And this means there is an inherent redundancy of about 30% in the system. So that is certainly a good first evidence that there is redundancy in our collection, and I see from other data that this is also the case, or would be the case in other collections, at least for cereals. However, this is certainly not the end of the story. We now need to start to the gene bank curators in order to initiate discussions on decision making. So what criteria need to be fulfilled in order to eliminate or to drop an accession from a collection. It's certainly not sufficient, or not only sufficient, to just look at the GWAS data. We need to again look at passport data. We need to look at phenotypic data. And then finally, we probably, at the end of the decision tree, we are able to make an informed decision. Well, genomic data, I'm running out of time, also provide very valuable information regarding the analysis of the representation and the distribution range of our collections. And this is just a principal coordinate analysis of those 15,000 barley accessions that have been GWASed so far. And what you see in this POCA is that the geographic patterns can be or are mirrored and reflected in at least the first two coordinates of this principal coordinate analysis. So that's certainly not really new, but the genetic diversity pattern mirrors the geographic origins of the material. However, to take this a further step on, we also can look at or answer the question, how deep and how well is the coverage of our collections with respect to other collections. And what you see here is a comparison to a part of the Swiss gene bank regarding these their barley accessions, and they are in this POCA marked in red. And what we can glean from this analysis is that the distribution space of the Gatersleben or IPK barley collection also would cover and include the distribution space of these 684 samples from the Swiss gene bank, and a very similar result was obtained when we compared the distribution range of our material, or the distribution space, with material that we have obtained from the gene bank of the Chinese Academy of Science, of Agricultural Science. So this is just a first approach, and now, of course, the next step is to compare the entire inventory of different gene banks in order to identify whether there are unique patches of diversities or of diversity kept in one or the other gene bank, or whether we are also all working in the same distribution space. Well, finally, the big challenge, of course, is data management. Data analysis, I think, is well taken care by the bioinformaticists. Data analysis usually is being performed in the ivory tower of science, and it's a bit disparate from the access of the data to the end users. And therefore, we are working hard on improving this end of the data flow chain. And we initiated the development of a web portal which will help to external users to inform the selection of plant genetic resources of our gene bank by just linking data and linking the analysis of the genomic and phenotypic data and the passport data with the actual physical or biological resource. Basically, this means you can run a POCA, for instance, using the data behind this web portal, and then you can select individual accessions from this POCA and immediately request these accessions from our gene bank ordering system online. So that would be the final stage once this portal goes online. Well, that takes me to the conclusion. Digital sequence information reveals an enormous potential in consolidating existing collections at the local, at the regional, and at the global level, along with the augmenting body of phenotypic information. This type and the genotypic information as well will help to inform the selection process of external users for plant genetic resources that are requested from GeneBank, and therefore the just described web portal is under development. And finally, the purposeful integration of high-value phenotypic and genotypic information with the biological material requires an extension of the management of plant genetic resources, because we have two types of collections. We have the accessions, which are frequently inhomogeneous. They are populations, land races, for instance. And we have the genotypes that were being used for phenotypic and genotypic analysis. So we will have, and we need to establish these genotype collections to really have a sustainable link between the biological material and the information that comes with it. With that, I want to close, but not without reminding you to mention a upcoming conference, the German Plant Breeding Conference, to which, on behalf of the German Association of Plant Breeding, I gladly invite you. The conference will take place in February and early March for a period of 3 days, somewhere in central Germany. And the topic really extends what we learned during and will learn during this meeting. It's about leveraging the value of genomic information. And we have a run-up or a lineup of very interesting speakers who will share their latest results with us. Thank you for your attention.
Thank you. And yes, I'll take one question before I hand over to my co-chair for the next two talks. One question, yeah.
I'd like to take your wisdom for a question I'm having. When you go for a phenomics work, you can have a sensor to the plant or plant to the sensor for evaluation of phenomics work. And most of the evaluation is plant to the sensor. The model you showed, the plant goes to the sensor to be evaluated by the sensor. You can, in the phenomics evaluation, either the plant goes to the sensor to be evaluated, or the sensor goes to the plant to be evaluated. Let me complete my. Yeah, the model you are showing, the model you are showing, it is a plant to sensor model, right? It is a advantage and it is a limitation of not mimicking the real situation. Similarly, when you take a germplasm for genotyping and you are mapping population for genotyping, both have advantage and disadvantage. If you go for very precise phenotyping versus phenotyping, so my question to you is, going for a very in-depth information for drought tolerance, which is a very tricky subject, should a plant to sensor is a better way of phenotyping or plant to sensor to plant is a better? Because sensor to plant will be more precise of field condition.
Yeah.
Similarly, plant population.
We may request you for having a discussions during the
drought is a very difficult issue. The experiment I described is plant to sensor, but in the plant cultivation hall, the huge phytodrone, we also set up a different system where the sensor moves to the plant and the containers where the plants grow are much larger. They hold about 1.5 cubic meters of soil substrate. So this is much more field conditions than we have in the normal usual greenhouse.
Thank you very much.